The idea that certain types of microbes can lead to cancer was introduced in 237 and 238. A selection from one of the online lectures is available on Blackboard as a review. A term that's sometimes used when discussing cancer is transformation. When we're talking about cancer, in this particular context, transformation refers to the changes of a host cell as the result of mutations or infection with an oncogenic virus. Changes that occur in human cells that can lead to cancers typically involve cell cycle control, and this may be direct or indirect. This is an overview of the cell cycle in human cells. Many pathways and mechanisms are involved in regulating the cell cycle. Signals include hormones, growth factors, and a number of external factors. Loss of cell cycle control results in uncontrolled cell division. As the name suggests, oncogenes are involved in cancer in many cases. In normal cells, we have proto-oncogenes, and these encode proteins that normally stimulate cell division when it's necessary and appropriate. In cancer, such genes may be altered, and in this state they become oncogenes, and this figure shows some of the different ways in which that can happen. There are several that are well known and associated with human cancers, including one called RAS. Viral oncogenes are also involved in promoting cell division, but these are introduced from carcinogenic viruses. The genes that are found on these viruses, though, are thought to have originated from ones in humans that were picked up by the virus. The products of proto-oncogenes accelerate cell cycle progression, while tumor suppressors serve as the brakes. At cell cycle checkpoints, DNA damage or other issues can be addressed. Tumor suppressors can arrest the cell cycle to allow DNA repair, or if DNA repair isn't possible, they can induce apoptosis or programmed cell death. This prevents cells from replicating when they have damaged DNA. Mutation of tumor suppressor genes or inhibition of tumor suppressors can result in loss of cell cycle control. Without these necessary controls on cell division, cells can multiply, and this can lead to development of tumors. P53 is an important tumor suppressor, and it's sometimes called the guardian of the genome. It plays key roles in pathways for DNA repair, differentiation, cell cycle arrest, and apoptosis in response to stress conditions. P53 is a DNA binding protein, and this allows transcription of genes in the various response pathways. The majority of human cancers involve a mutant or defective P53, and this is usually a P53 that cannot bind to DNA. Some viruses have genes that encode P53 inhibitors. While most human cells have limits on the number of times they can divide, cancer cells are not subject to these limits. They are, in effect, immortal. To do this, they have to maintain their telomeres on the chromosomes, and these are just repetitive non-coding sequences that are found at the ends. There is a human telomerase enzyme, but it's not active in most cells. Typically, we only see it working in germ cells, stem cells, and the majority of cancer cells. In DNA replication, DNA polymerase can synthesize DNA only in a 5' to 3' direction. With linear chromosomes, DNA synthesis cannot continue to duplicate all the way to the end of the chromosome, and this means that the end of the chromosome is shortened slightly each time. The reason for this is that the Okazaki fragments require RNA primers ahead on the lagging strand. During replication, the RNA primers are gradually removed until you only have primers at the very ends of the molecule. Once those are removed, a gap remains. At the end of the chromosome, there's no nucleotide sequence in the 5' direction, so DNA polymerase can no longer add any nucleotides, halting synthesis. This leaves a small portion unreplicated. Telomeres at the ends serve as buffers. During cell division, portions of the telomere will be lost, but this doesn't harm the cell. Having disposable DNA at the end of the chromosomes protects the genes before them, and this prevents them from being truncated, at least until the telomere is gone. Human cells without telomerase, which is most of them, gradually lose sequences from the telomere ends with every replication. As the telomeres shorten, our cells eventually reach their replicative limit. Telomerase is a type of reverse transcriptase and is able to add telomere units to the ends of chromosomes. Cancer cells often become immortalized by activating telomerase, and this extends the telomeres of the chromosomes in the cell. Telomerase consists mostly of protein, but it also includes a single molecule of RNA that contains the template for building the telomere subunits. When adding to telomeres, telomerase orients the tip of a strand of DNA onto the RNA portion so that the template is adjacent to that DNA tip. The enzyme then adds and links DNA nucleotides until the full subunit is formed. Once that subunit is done, telomerase can attach another one by moving to the new end of the chromosome and repeating the process. The idea that some cancers have an infectious cause may surprise some people, but enough evidence has been gathered that expert panels of the International Agency for Research on Cancer have classified several major infectious agents as human carcinogens. These include a number of viruses, as listed here, along with the bacterium Helicobacter pylori. Evidence linking these viruses and H. pylori to cancer was gathered from numerous epidemiological, clinical, and basic biological research studies. It's difficult to provide an accurate measure, but it's estimated that more than 15% of cancers can be attributed to infections worldwide. It should be noted that the rates are much lower in developed nations than in developing countries. Also, as screening and diagnostic measures improve, it's possible that additional cancers and cancer-causing microbes may be identified. Before we take a closer look at some of the specific pathogens and cancers, there are some general points about infectious cancers to think about. If we look at the general mechanisms that are involved, there are two that are considered really important. These include the fact that chronic infection leads to chronic inflammation, and the fact that in some cases, viral genomes can integrate into the host genome. In addition to those mechanisms, certain viruses, like HIV, have effects on the host immune system, and this can reduce the ability of the host's immunosurveillance capabilities, which are very important in preventing cancer. This loss of immunosurveillance leads to not only increased risk of cancer, but also increases the risk of very aggressive cancer occurring. The listed infectious carcinogens have the ability to cause persistent or chronic infection in at least some individuals. These infections induce chronic inflammation, which is known to be associated with cancer development. During chronic inflammation, the recruitment of phagocytes leads to increases in reactive chemicals, like reactive oxygen species, or ROS. These can damage DNA as well as other cellular components. The damage in DNA can lead to changes that promote carcinogenesis. Also, with the constant or repetitive cell damage, cycles of cell proliferation often occur, and this gives more opportunities for DNA damage and potentially allows cancerous cell growth. In some viral infections, the viral genome can insert into the host genome. This can interrupt host genes involved in cell cycle regulation and could also introduce viral genes that are oncogenic, allowing promotion of cell division or possibly interfering with tumor suppressors. Although there is considerable variety in the known carcinogenic infectious agents, some common features exist. One major common feature is the fact that each of them has the ability to cause chronic infections. Additionally, many of these pathogens are highly prevalent within the human population, even though the cancers that they cause can be quite rare among people with these infections. Often, cancers only occur after many years or sometimes decades after the initial infection. In many cases, if a virus is causing cancer, the genome of that virus would be present in the malignant cells. And other factors typically play a role in whether or not an infected individual actually develops cancer. The age of infection appears to play a role in some types of cancer, where a very early age of infection is more strongly associated with the risk of developing cancer. Factors that affect the host's immune state, things like nutrition and other aspects of health, are also likely to affect the risk of malignancy. Given that infection doesn't always lead to cancer, and that there's usually a long delay before cancer occurs, it can be difficult to definitively link a pathogen to cancer. Just because a virus or other microbe is present in tumor tissues does not necessarily indicate a causal relationship. In addition to epidemiological studies showing a strong relationship between infection and cancer, in vitro studies should also allow some view into the mechanisms by which an agent induces cancer. Infectious cancers, where a person becomes infected with a virus or bacterium that can lead to development of malignancies, are different from what are called contagious or transmissible cancers. Contagious cancers occur when cancerous cells from one individual are spread to another, leading to malignancy in the recipient. In humans, this appears to be quite rare, occurring in unusual and sporadic cases. However, in some other animals, these types of cancer can move from individual to individual by direct transfer of cancerous cells, and this happens quite regularly. An example would include a sexually transmitted cancer in dogs, as well as the devil facial tumor disease, or DFTD, that occurs in Tasmanian devils and is spread through facial bites during fighting. An additional tumor disease was also discovered in Tasmanian devils, which makes us wonder if they're uniquely susceptible to contagious tumors.